Welcome once again. So today I'm going to give you some basic information about cancers that will help you form an idea of what cancers are. All right. So let me share the screen with you and then we'll start with the lecture. Okay, here it is, share screen. All right, an introduction to cancers. So let me start by telling you that cancer is not one disease. Every cancer is different from another cancer, all right? We have about 200 different types of cancers, but even in one type of cancer, let's, for example, take breast cancer. A breast cancer in one woman might be different from the breast cancer in another woman if you go into the details of the genetic mutations and the molecular markers of that cancer. All right. So the first thing is that cancer is not one disease. Cancers are many diseases that share one common feature. And that common feature is growth dysregulation, which means that there is no control on cell division. The cells of the cancer or the cancer cells, they go on dividing. They cannot be stopped. So that is what the basic problem is. Why are the cells dividing and dividing? Well, there could be different reasons, but the basic reason is something has gone wrong with the genes that regulate the cell division, okay? So some cancers are highly treatable, like Hodgkin lymphoma, and unfortunately, some cancers are highly fatal, like the cancer of the pancreas. Now, just a few nomenclature. I'll give you some more clinical terms in another short lecture, but let's start with the very basic nomenclature of the cancers. Oncology, I'm sure you all know, it is the study of cancers or the study of tumors. Neoplasia means new growth. Tumor is a lump or a mass of abnormal tissue or abnormal cells that resembles a swelling. A benign tumor is the one, obviously, it's not a cancer. It remains localized and you can remove it by surgery. You can resect that very cleanly if you want to remove it, all right? Transformed cells are the cells that continue to divide, and in other words, the cancer cells are transformed cells. Malignant neoplasm, it means a cancer. It grows, it invades adjacent tissues, and it can even spread to distant sites. Metastasis, which is spread of malignant tumor to other organs, such as bones, lung. For example, breast cancer can metastasize to bones, which is pretty common. It can metastasize to lungs, brain, liver, and so does the colorectal cancer or any other prostate cancer. All these can metastasize or they can invade other tissues which are close by, all right? So what is a cancer made of? This is very important because when you come to the treatment of cancer, you will see that we have got many different types of treatments, but a cancer is made of two things. Like all the other tissues, the transformed cells, they form the parenchyma of the cancers. These are cancer cells that keep dividing. You have parenchyma in every organ in the body, and you have connective tissue in every organ of the body. That connective tissue is also known as extracellular matrix, all right? So the cancer cells have parenchyma and the cancer cells have supporting cells, which are called the stroma. These are non-cancer cells. They keep cancer cells together like the connective tissue in normal organs. But they're very important because stroma has blood vessels. It has got inflammatory cells in it. And you know, blood vessels, as the cancer grows in size, it needs more oxygen, it needs more nutrients. And how will it get all those nutrients and oxygen? Through the blood vessels. So stroma contains the blood vessels. Without stroma, the cancer cannot grow, which means if you target the stroma in the treatment of cancer, you can eliminate the cancer. Actually, that is one of the treatments that we give in combination with the drugs that actually kill the cancer cells, okay? Benign tumors, they do not spread. They are not cancers. They are named by attaching an oma to the tissue from which they arise. For example, fibroma arises from fibrous tissue. It's not cancer. It's just an overgrowth of the fibrous tissue. Chondroma is cartilage. When a cartilage becomes bigger abnormally, we call it a chondroma, but it's not a cancer. Adenomas means something related to the gland. We can have two types or two phenotypes of adenomas. They arise from epithelial cells and form gland-like structures, or they can arise from glands, and but they lack glandular growth pattern. So either they arise from a tissue like the epithelial cells and they form gland-like structures, or they arise from the cells of a gland. For example, they can arise from the adrenal gland, the adrenal cortex, which is a gland, but they lack the shape of a gland, okay? Papilloma means a finger-like growth from the epithelial tissue. Polyp is a growth that projects above the mucosa in the GIT. It's like a balloon attached to a string, all right? So they are very common, not very common, but they're quite dangerous because they can transform into cancer, especially in the colon. Nasal polyps is a term that you might come across. Nasal polyps are different. They are inflammatory in origin. They are not benign tumors, or they are not tumors at all. All right. Now, malignant tumors, which are not benign because benign tumors do not spread, and but the malignant tumors, they invade and they spread. We can have carcinomas, which are very common. We can have sarcomas. We can have leukemias, and we can have lymphomas. We can also have tumors of the nervous tissue, all right? So carcinomas develop in epithelial cells lining the internal organs, or they cover the outer body structures. You know, epithelia has got many different origins. When I say origins, I mean the embryological origins. They can have cells from the endoderm, from the mesoderm, from the ectoderm, all right? So the cancers that arise from the epithelial cells, they are known as carcinomas. Sarcomas develop in mesenchymal cells that form connective tissue, bones, cartilage, muscle, blood vessels, fat. Mesenchyme is connective tissue in the developing embryo, and mesenchyme is a part of mesoderm. Leukemias are the cancers of the blood cells, and leukemias and lymphomas are both the cancers of actually immune cells, okay? Cancers of lymphatic system, these are solid tumors. They are commonly B cancers of the B lymphocytes, okay? Most commonly, rarely we can get lymphomas from T lymphocytes, but most commonly they arise from the B lymphocytes. Now we have two types of carcinomas. We have got adenocarcinoma, which grow in glandular pattern. They are more common. We can have squamous cell carcinomas. They produce flattened cells, which are known as squamous cells, all right? Now we can have well-differentiated cancers, and we can have poorly differentiated cancers. This is something very important, because when you want to predict the aggressiveness of a cancer in your clinics, when a cancer patient comes to you, you look at the stage of the cancer that we will come to later on when we do actual cancers, and you also look at the aggressiveness of the cancer. We call it the grade of the cancer. So well-differentiated cancers are less aggressive. They grow slowly, and poorly differentiated cancers, they grow and they spread very fast. But this one has a drawback as well. You know, slow growing cancers are detected, I mean, they are not detected sooner, they are detected a bit late as compared to poorly differentiated cancers. So in well differentiated cancers, the tissue of origin can be identified because the cancer cells, they are well differentiated and they look like the cells of the organ from which the cancer originated. But in poorly differentiated cancers, the cells are so abnormal, the cancer cells do not look like the cells of the organ from which they arose. They are also called undifferentiated cells. So just by looking at the cells, you cannot tell from which organ they came from. For example, let's look at this building. This is the original building, and this is the building after it raised or something happened to this building. If you just look at this building, you can never tell that the original building was something like this. Although from the surrounding structures, you might be able to tell, but if I give you just some rubble from this building, you won't be able to tell me that it came from this building. So this is what a poorly differentiated cancer looks like. And here is a well differentiated, uh, I mean, this is analogous to a well differentiated cancer, although this building has changed a lot, but you can still recognize by looking at this building, these structures. Or here, you can tell that this building came from this original building. All right, so this is well differentiated, just like this. When the pathologist gets the cancer cells, he gets a biopsy, he looks at the cell, and if he's able to tell which organ the cells came from, then they are well differentiated. Although he will know because the surgeon who has taken the biopsy will tell him that he got the biopsy from which organ, but the cells sometimes look so different that they do not resemble the original cells in any way. All right, so that's poorly differentiated and well differentiated. Three features that make a cancer dangerous. Poorly differentiated cancers, they are more dangerous than well differentiated cancers. Local invasion, penetration of cancer cells into surrounding tissues, like the roots of a tree infiltrate the ground. If the cancer cells have penetrated into the adjacent organs, then it's more dangerous. And metastasis, spread of tumor to different organs. All right. Another term is anaplasia, which is sort of synonymous to poor differentiation. Cancer cells are differentiated morphologically and functionally. Cancer cells are different morphologically and functionally from the cells of the tissue of origin. Morphologically means in shape, and functional means in their working. All right. And Local invasion, we have another term which is carcinoma in situ. This is a term used for dysplastic cells, not cancer cells. What is dysplasia? I hope you remember from your pathophysiology. Hyperplasia, metaplasia, dysplasia, dysplastic cells. They are disorganized. They have different shapes. They have different sizes. Normally in the epithelia, the cells are dividing only in the basal layers, but when the cells become dysplastic, you'll find dividing cells in all the layers of the epithelium. This is known, we call them mitotic figures in all the layers of the cells, okay? And it involves the entire thickness of the epithelium. Now, this is not cancer, although there is a bit of a disagreement among different people. But we'll come to this once again in the next slide. And metastasis spread to distant organs. About 50% of the cancers, when diagnosed, have already metastasized. That is why, we want to screen people, older people, women after the age of 40, 50, 60 years for different types of cancers, all right? Because cancers, the early you diagnose a cancer, the better it is. The greater the chances are for getting a cure of the cancer. So 50% have already metastasized, 30% are clinically evident. You can diagnose them or you can find them, but 20% are occult, they are hidden, you can't see them. So the patient will not go to the hospital. Why would a patient go if he doesn't suspect any disease in his body? So that's the problem. And that is why, many insurance companies, they want you to have certain tests uh, to, I mean, to discover or to find if a cancer is growing in your body, all right? So you might come across disagreement or classification of carcinoma in situ, but I'll just show you what carcinoma in situ is. Some people call it dysplasia, as I explained to you. Others call it precancers. Still other call it a cancer, all right? So this is how it starts. This is the epithelium and this is the basement membrane. These are the epithelial cells, and we see that one cell has mutated and transformed. Transformed means that the cells, this specific cell has lost control over cell division. So it will divide into 2, 4, 8, and so on. So this cell divides, and initially it is hyperplastic. All the cells are about the same. You can recognize them, same size, same shape. But then they become dysplastic, right? They've got different shapes. The nuclei are bigger. We often look at nuclear to cytoplasmic ratio. So compared to the cytoplasm, the nucleus becomes very, very big, all right? So this is one of the uh, markers of dysplasia. And then we get carcinoma in situ. Now, note up till now that the basement membrane is intact, although you see that one cell has sort of tried to penetrate into the basement membrane, but the basement membrane is intact. So we call it carcinoma in situ. Now, what you see over here is that the basement membrane has been invaded, it has broken, and the cancer cells have gone down into the submucosa. And what you find in the submucosa are blood vessels, so the cancer cells can enter the blood vessels and they can go to other parts of the body and they can form secondary cancers, all right? So this is what carcinoma in situ is. Dysplasia, if you remove the trigger or the irritant, the dysplasia is still reversible, but some people believe that carcinoma in situ is not irreversible, all right? Okay, so here it has broken through the basement membrane. Now, local invasion, as I explained to you, the cancer cells can penetrate into the adjacent organs, like, this tree. This tree is present above the ground, and it has got roots down below the ground, okay? If you just remove the tree, these roots will still be there. So infiltration into and destruction of the surrounding tissue. Now, you see, that is why when a surgeon operates upon a cancer, he does not only remove the tumor, right? Let's assume that this tree is the tumor. It has got its roots in the ground. So you have to, if you want to completely remove this tree, leave behind no signs of the tree, you have to remove a lot of the ground as well, okay? So that's what the surgeon does. The infiltrative mode of growth makes it necessary to remove a wide margin of surrounding normal tissue. So when a surgeon removes the tissue, he will remove a lot of normal tissue as well, and then he will send all that removed resected mass to the pathologist, okay? Surgical excision of a malignant tumor is attempted. So he removes a lot of normal tissue. Why? He sends it to the pathologist, and the pathologist is going to look at the margins of the tissue that they have received, okay? Surgical pathologists carefully examine the margins of the resected tumor to ensure that they are devoid of cancer cells. For example, if you remove the tree, you must remove all this ground so that all the roots are gone and you don't find anything here. Let us suppose that and these will be the margins. So what will you find in the margins? Just the earth or whatever material is there under the ground, okay? But let us suppose that if you do not remove adequate amount of roots, then if you examine the margin, you will be able to see the roots, which means some roots have been left behind in the soil or in the ground. Okay, the same happens in the cancer. You must remove a lot of extra tissue, and when the pathologist examines that tissue, no cancer cells should be there in the margins of the resected tissue. Now, metastasis, I've already explained to you, it is defined by the spread of tumor to sites that are physically discontinuous with the primary tumor. For example, if the tumor is in the lung, it might go to the spleen, or it might go to bones, to the vertebral column, it might go to the brain, which is not connected physically to the lungs, okay? The invasiveness of cancers permit them to penetrate into blood vessels, lymphatics, and body cavities, providing opportunities for spread, right? So basically, they might spread from blood vessels or through lymphatics as well, or they can, as I'll show you, something known as this as seeding that I will explain you in a little while. 30% of patients with newly diagnosed solid tumors present with clinically evident metastasis, as I explained previously, and an additional 20% have occult or hidden metastasis at the time of diagnosis. In general, the more anaplastic and the larger, now anaplastic means poorly differentiated, and the larger the primary neoplasm, okay, the size of the tumor, the bigger or the size of the tumor or the cancer in the tissue, the more likely it is to metastasize. Now, over here you can see that the primary cancer has metastasized to the liver, to the brain, to the kidney. Actually, the primary cancer was in the liver, but it has metastasized to the brain and to the kidneys via blood and the lymphatics, okay? And again, poorly differentiated cancers are more likely to metastasize. Okay, spread of cancers, spread by seeding. I'll give you examples. Seeding occurs when neoplasms invade a natural body cavity. For example, cancers of the ovaries. For ovaries, they lie in the peritoneum. So if there is an cancer in the ovaries, ovarian cancer, the cells from the ovary might go to some other part inside the peritoneum. So this is known as the seeding. Okay, it has gone a little bit away from the ovaries, but it is within the peritoneum. It has not gone through the lymphatics. It has not gone through the blood vessels. Another example is the neoplasms of the CNS, such as medulloblastoma or ependymoma. They may penetrate cerebral ventricles, the lateral ventricles, the third ventricle, the fourth ventricle, that are all connected to each other, and they may be carried by the CSF to the meningeal surfaces that could be in the brain or in the spinal cord. So what you're seeing over here is they have spread to non-adjoining tissues or areas. They have gone a little bit away from the primary cancer, but not through lymphatics, neither through lymphatics nor through the blood vessels, okay? So this is what seeding is. Spread via lymphatics. Carcinomas commonly spread via lymphatics. So that is carcinomas. Now, what do sarcomas do? Spread via blood vessels. Sarcomas commonly spread via the blood vessels, but you know that there are connections between the lymphatics and the blood vessels. The lymphatics, through thoracic duct, they drain into the blood vessels, into the subclavian vein, all right? So they're interconnected. Lymphatics and blood vessels are interconnected and used by both types of cancers. Sentinel lymph node is an important term that you must understand. This is the first lymph node that receives lymph from the primary tumor, from the tumor mass. Every organ has got lymphatics, which drain into the regional lymph nodes. So the first lymph node, there are ways to find that out. You inject something into the cancer while you are operating upon the patient, some sort of colored chemical like India ink, and you see where that color goes. It will go through the lymphatics to a particular lymph node, and that would be the lymph node, one lymph node or more than one lymph node. So they are known as sentinel lymph nodes. And mysteriously, cancers rarely metastasize to skeletal muscles. Anyway, all right. So this is what it is showing. I mean, I'll not go into details. Lung carcinomas arising in the respiratory passages metastasize first to regional bronchial lymph nodes and then to tracheobronchial. So the any particular area of the lung or any other tissue will drain first into the nearby lymph nodes, which are known as regional lymph nodes. They are very close to the organ, and then from there they will go to the next level of the lymph nodes, and in the lungs they are known as tracheobronchial or hilar lymph nodes. Okay, so we also call them N1 or N2 nodes, which are a little farther from the region which is training into them. Right. Epidemiology. According to WHO, due to increasing population size, the number of cancer cases and deaths will increase worldwide, and that makes sense. But the problem is, as the population increases, the burden of cancers are increasing, and the health system is sort of being overwhelmed by the cases. That's why we say that we must do screening, although that is also not possible. It needs a lot of resources to screen the whole population. Screening just means that we look for certain clues, certain markers in the blood that might indicate that a cancer might be growing somewhere in the body. All right. Right. So as the population size increases, the number of cancer cases will also increase. In the USA, the incidence of cancers in men and women has been stable since 1995. All right. The death rate has decreased in the USA. Now, why is that? 20% decreased in men, the death rate, lung, prostate, colorectal cancer. 10% decreased in women, breast and colorectal cancer, and that has been attributed to screening, right? Also to the decreased use of tobacco, and improved detection and treatment. Detection means screening. For example, women are advised to do mammography, which is a sort of an X-ray of the breast, okay, after a certain age. At every year we'll come to that when we do breast cancer, okay? So whether they have cancer or not, they are also advised to do self-breast examination, so that if they find any lump in their breast, they immediately go to the doctor and they get further investigations for that lump, all right? So improved detection and treatment is an important cause for the decreased death rate in the USA. Now let's go on to the known environmental factors that are linked to cancer. Diet, red meat is the culprit, okay? Increases the risk of cancers, and high fiber diet reduces the risk of cancers. Vegetables, fruits, cereals, these are good for, they prevent development of cancers, all right? Smoking is a risk factor for many cancers, particularly lung cancer, but many other cancers can develop as a result of smoking, or you can say that smoking has been implicated in many types of cancers. Why? Because cigarette smoke has got about 200 to 300 carcinogens, which means chemicals that can produce cancers in the body, right? Alcohol causes cirrhosis of liver, and it can lead to cancer. Infections can lead to cancers. I can give you the example of hepatitis C virus, that could lead to hepatocellular carcinoma. I can give you the example of H. pylori infection in the stomach that could lead to carcinoma of the stomach. Epstein-Barr virus can cause Burkitt's lymphoma. So infectious agents are also a cause of cancer, all right? So environmental factors contributing to cancers, there are so many of them: pollution, alcohol, occupation, infection, reproductive sexual behavior, foods, medicines, diet, whatever. But the two most important are tobacco and diet, all right? Now, different types of cancers. These are statistics from USA 2016 statistics. This side shows the incidence of cancers, and this side shows the mortality or the death due to cancers. And there are so many different types of cancers, but I'll show you only a few which are the most common type of cancers. Lung cancer, it's not the most common type, but look at the death rate. The incidence is 14% in men and 13% in women, but the death rate is about 27% in both men and women. And if you look at prostate cancer, here the incidence was 14%, but here prostate cancer, obviously it will be there only in men, but the death rate in prostate cancers is pretty low, 8% as compared to 27% in lung cancers, right? Similarly, breast cancers, overwhelmingly it is in women, very rarely. It could be there in men as well, because breast tissue, although very rudimentary, breast tissue is present in men as well. And it develops in women because of estrogens. So 29% incidence, and the death rate is about 14%, all right? And colon cancer incidence is 8%, and the death rate is also about 8%, right? So these three are important cancers, and we are going to do these three cancers when we come to therapeutics. Other factors that predispose to cancers: age. Most cancer deaths occur between 50, 55, and 70 years of age. Cancer is a disease of old age, all right? The major lethal cancers in children are leukemias and lymphomas. Chronic inflammatory conditions may predispose to cancers. For example, ulcerative colitis, and I'm going to give you more examples as well. Immunodeficiency states increase the risk of cancers. HIV/AIDS does cause many cancers. Even, post-transplantation patients who are on immunosuppressive drugs, they have a higher incidence of cancers, because immune surveillance eliminates a lot of cancer cells. As soon as they become transformed cells, they can recognize them. Natural killer cells, they are always looking for cancerous cells, and they kill the cancerous cells. So immunodeficiency states can increase the incidence of cancers. Precursor lesions. Precursor lesions are associated with elevated risk of cancers, right? I've already mentioned many to you. They commonly arise secondary to chronic inflammation or hormonal disturbances in endocrine sensitive tissues or hormone sensitive tissues, like the breast, like the prostate, like the ovaries, like the uterus. They're all hormone sensitive tissues. The most common are seen in habitual smokers. Habitual smokers, they always have some inflammation in their airways, which is known as chronic bronchitis. So itis means inflammation, and that is a risk factor for lung cancer. Endometrial hyperplasia and dysplasia due to unopposed estrogenic stimulation. We have done that in contraception as well. We saw that women who have uteri, they are always given combined oral contraceptives, because we cannot give estrogens. Estrogen has an effect of cell division, right? They cause the cells to divide. So we combine them with progestogens to decrease the risk of cell division. Okay, so if you just give estrogens, there is a risk of endometrial carcinoma. And previously in the lecture for antineoplastic drugs, you have seen a drug tamoxifen, which blocks the estrogen receptors in the breast tissue, but it is an agonist in the endometrium. So there is a risk of developing endometrial cancer when tamoxifen is used for a long time. Okay. All right. Then another example is leukoplakia, which are thick white patches due to chronic irritation in the mouth. Okay. They could be there in the vulva, on the penis as well, and they could progress to squamous cell carcinoma. Villus adenoma of the colon, which is a type of a polyp. I mentioned that the polyps are high risk factors for development of colorectal cancers or colorectal carcinomas. Right, now here is a chemical carcinogen, aflatoxin B1, derived from fungus, right? So fungal infection means inflammation, and this toxin is also going to cause damage and initiate an inflammatory response, which contaminate this aflatoxin. It contaminates grains, food, and it most commonly induces hepatocellular carcinoma. All right. So let me remind you that cancer is not one disease. Cancers are many diseases that share one common feature, and that common feature is growth dysregulation. Causes of cancer. Cancer is a genetic disorder caused by DNA mutations. These mutations are induced by mutagens. What are mutagens? Mutagens are factors that cause mutations in the DNA that bring about certain changes, detrimental changes in the DNA that I'll explain to you in a little, that could lead to cancer. Okay, smoke is one of them. Aging is another. Radiation is one of them. We have seen aflatoxin A in the previous slide, wherever it was. Okay, the aflatoxin B1, sorry. So these are known as mutagens, or you can also call them carcinogens. I said that smoke has got 200 chemicals that are carcinogens, all right? Cancers also show epigenetic changes. Now, epigenetic, in the genes, we can have two types of changes. Either we can have a change in the sequence of the nucleotide. DNA is made of adenine, guanine, cytosine, thymine, whatever the arrangement is. But if we change the sequence, that is not an epigenetic change. In epigenetic change, the sequence remains the same. Something else happens. And one of the things is DNA methylations or histone modifications. All right. Those of you who have not done genetics, they might find it difficult to understand what I mean by this, but I'll try to explain that to you. So let's start with mutations. I've said that there are mutations. Cancer is a genetic disorders caused by DNA mutations. Now, mutations are of two types. One is a change in the sequence of the DNA that I'll show you over here. What are mutations? Before that, let me remind you what DNA is. DNA is a sequence of 4 bases, thymine, guanine, adenine, cytosine, and they go on and on. There are millions and millions of these bases, okay? Adenine, guanine, cytosine, thymine. In the messenger RNA, I hope you know that in place of thymine, we have got a uracil, right? So this long chain of DNA is compacted. There are many levels of compaction of DNA, and then they make the chromosomes, and chromosomes have got different parts, centromeres, telomeres, or whatever, okay? But the thing is that in the whole genome, we have got 23 pairs of chromosomes. We have got about 6.4 billion nucleotides in the diploid genome, all right? So this is the sequence of DNA. Single nucleotide polymorphisms, which are also known as SNPs. Now, this is something normal. It's not a mutation that would lead to cancer. We do call it mutated sometimes, but it doesn't mean that it will lead to cancer, right? For example, one person in a certain segment of the DNA can have this sequence, thymine, adenine, guanine, guanine, cytosine, adenine, cytosine, thymine, guanine. Another person will have the same sequence, but in place of an adenine, he has a guanine, right? So this is known as single nucleotide polymorphism. This person has got A at this place. This person has got a G at this place. This is something normal, okay? It doesn't mean that it's going to cause cancers. We have got millions of single nucleotide polymorphisms. Approximately after every 1,000 base pairs, we get a polymorphism in different people. And it is because of these polymorphisms that every person is different from the other person, different in color, different in body weight, difference in body weight, difference in height, whatever, okay? It's because of these polymorphisms. Identical twins have exactly the same genome, exactly the same sequence of DNA. They do not have any different single nucleotide polymorphisms, so they are exactly alike, okay? They are identical, right? So I'll show you the types of mutations. This is an original sequence, okay, whatever it is. So what happens is that these three bases, they have fallen off, okay, due to some reason, maybe due to radiation, maybe due to free radicals. The DNA bonds have been broken and this has fallen off. We call it a deletion. So this has gone away and the remaining portions of the DNA will come together and we get a deletion of this thing. You can have deletion of 3 base pair, 1 base, 2 bases, bigger piece of chromosome, all right? So these are known as deletions. Here's another type of mutation. This is the original sequence. What has happened is that the original sequence, TGC, has been replaced by another sequence, GAT. This is known as substitution. Again, you can have a substitution of 3 nucleotides, 1 nucleotide, 2 nucleotide, all right? So this is what I'm going to show you. So we have two terms, a wild type and, alleles. If you have studied your genetics, allele is a form of a gene and different people have got different alleles. We get, in our genome uh, for every gene, we get one from the mother and one from the father. So we can call that allele of the gene, one allele we get from the father and one allele we get from the mother. So based on these polymorphisms, we call them wild type or mutant. Wild type, the type of allele or the type of sequence of the DNA, which is found in the majority of the population. And the other sequence, which is not the sequence that I have shown you, it might not be harmful. It may or may not be harmful, but we call that a mutant allele. Okay. Anyway, so what I'm going to show you is what type of mutations or polymorphisms we can get. Okay. See, this is a substitution. No, this GC has been replaced by a TA. The other GC and TA, are there, but this has been replaced by TA, so we call it a substitution. What has happened over here is that this GC is gone. We have got this TA, this GC, but this red one is not there. We call it a deletion. Here is an insertion. After TA, we have got a different nucleotide. The GC is there. This GC is there, but we got an insertion. Now, all of these are going to cause changes in the protein. If they are in the coding region of the gene, they might change the sequence. Again, may or may not. That I'll show you in a later slide. All right. For example, this is a wild type protein, which is, or you can call a wild type DNA sequence, which is making a protein. All right. So this proteins are big. They might have a few amino acids, or they might have hundreds of amino acids in them. So we are looking at amino acid number 211 on the polypeptide chain, which has been coded by the DNA. Now, this is a glycine, and the codon for glycine is GGA. I hope you know what a codon is, all right? Now, whenever you have this sequence on the DNA, you will make a glycine. But what happened was that this G was replaced by an A. Now, AGA is not the sequence for a glycine. It is a sequence for an arginine. So if this mutation is over here, then in place of a glycine in this protein, whatever protein it is, we will get an arginine at position 211. Originally, or we should have a glycine, but because of this mutation, we got an arginine. Well, let us suppose that this G is there and we change or substitute this G with an A. Now we have got GAA in place of GGA. What does GAA make? It makes a glutamic acid. So if this mutation is there, then in place of glycine in this polypeptide chain, we will get a glutamic acid. And sometimes it has got catastrophic consequences. For example, you all know the disease sickle cell anemia or sickle cell disease. In the gene for hemoglobin, the O2 carrying molecule, that's what hemoglobin is, a sickle cell mutant caused by a single nucleotide shift. Okay. So what happens is, this is the normal hemoglobin DNA at position 6 of which means the sixth amino acid. This is normal. We have a CTT. This is the codon for glutamic acid. Okay. This is the DNA, and this is the codon on the messenger RNA, guanine, adenine, adenine. So whenever there is a guanine, adenine on the messenger RNA, we will get a protein which will have a glutamic acid at this place. Okay. The transfer RNAs have got anticodons, if you remember from your cell biology. Anyway, but what happens in sickle cell anemia is that this T on the DNA is replaced by an A. Now what we get is CAT. And in the messenger RNA, which is complementary to the DNA, we get a GUA, a uracil. In place of an adenine, we have got a uracil. Okay, here it was guanine, adenine, which coded for glutamic acid. But what we got after the mutation was guanine, uracil, adenine, which codes for a valine. Just one amino acid change, and we get sickle cell hemoglobin. All right, and we have already done sickle cell disease in pharmacotherapy 3, and you know the consequences and the complications that we get in the sickle cell disease. Why? Just because one nucleotide was substituted. Okay, it was replaced by another nucleotide. So it has got very detrimental consequences, and we call it an E6V substitution. E stands for glutamic acid. At position 6, we get a valine in place of glutamic acid. Okay. These are examples of some other mutations. You can read it on your own when I give you the handout. Genetic alterations in cancer cells are passed on to the daughter cells. Now, some mutations provide a survival advantage to the cell. Now, we know that the cancer cells are dividing cells. Okay, they are rapidly dividing. Why do they go on dividing? Because they develop some mutation that gives them a survival advantage. And that mutation causes them to divide again and again. That is, a cell with certain types of mutations have greater potential for cell division and survival as compared to normal cells. Okay, I'll show you the normal growth pattern as well. But what I'm trying to tell you is that there has been a certain mutation in a gene. Cell division is also controlled by genes. And there are genes that promote cell divisions. There are genes that stop cell division. Okay. If a mutation occurs in any of those genes, either in cell division promoting genes, we can get repeated cell divisions. We lose control over cell division, and the cells go on dividing and dividing. All right. Cell with certain mutations will go on dividing and will form a large colony of identical daughter cells, because whatever mutation was there in the parent cell will go on to the daughter cell as well, and the daughter cell will also go on dividing, and it will keep going on. It will pass from one generation of cells to the next, and that is what a cancer is. It goes on dividing. We call them clones. All the cells of the cancers are identical, but sometimes, as the cancer is progressing, the cells can change themselves. Therefore, cancers are clonal and progressive. Progressive means that they keep growing. All right. But as the cancers progress, the clones can get more mutations, and they can, they have, that too has certain consequences, especially when we are giving treatment for the cancers. Now, cell division is a tightly controlled process. Originally, cells divide according to a program in the DNA. The baby starts with one cell. The ovum is fertilized by a sperm, and we get one cell. Okay. Now that cell goes on dividing and dividing, and it makes a baby. Cells divide when cell division is needed and stop dividing when cell division is no more needed. We start as one cell that divides and divides and divides according to a genetic program, and that programming is in the DNA, but the cell division is very organized. Okay, it's not like a cancer. Cancer has got no shape, but an ovum um, divides in a very organized manner. It makes different organs, gives shapes to the organs. Cancer cells cannot do that. Okay. Now, this is what from an embryo, which we get from a single cell, and they divide and divide, and over 40 weeks we get a normal baby. 99.9% of the times we get normal babies. All right. And after birth, body tissues grow by increasing the number of cells. Cells divide and organs grow very quickly between conception and adulthood. Now, everything grows. The teeth grow, the heart grows, the liver grows, even the eyeball grows. Everything is growing. The nails grow, okay? Everything is growing in the body. A stage comes when adult size is reached. Now, you see, these are the adult ladies, and now at this stage, all organs stop growing. Cell division is going to be there, but that is for maintenance and for repair of the organs, or before they replace the worn out cells. No more increase in the number of cells here until a person reaches adulthood. The number of cells increase, but once he reaches an adult size, the number of cells do not increase. In adults, most cells divide for the purpose of tissue renewal rather than for growth, okay? But in old age, the number of cells decreases, and that is why, in the brain, the person gets Alzheimer's, the function of the heart goes down, the immune system declines in function. So that is all a programming in the DNA that happens in every person and in every animal, okay? So what I'm trying to tell you is that cell division is a very tightly controlled process. Cells will divide according to a program. They will divide only when needed, and they will not divide when it is not needed. What happens in cancers is that we lose this control on cell division, and the cancer cells go on dividing, okay? Now, let's have a look at the cancer genes. Cancer occurs because mutations alter the function of genes. Mutations occur by the action of chemicals, radiation, viruses, and sometimes they can occur spontaneously. Some mutated cancer genes may be inherited. We have got many examples of that in breast cancer and colorectal cancer. Now, there are four major classes of cancer genes. We have got oncogenes, we have got tumor suppressor genes, We have got genes that regulate apoptosis, and we have got genes that regulate interaction between cancer cells and the host cells. Now, all these genes are present in normal people as well. And these genes are controlling the normal cell division. When these genes function normally, cell division will be normal. But when something goes wrong with these genes, when there is a mutation in these genes, then we lose control over cell division and we can get cancer. Okay. So cancer is basically a genetic disease. Now, proto-oncogenes are normal genes that cause cells to divide in a controlled way. So we all have proto-oncogenes. We all have tumor suppressor genes. We all have genes that regulate apoptosis, but they are working normally and they're working to keep us healthy, all right? They are helping to prevent development of cancer. But when proto-oncogenes mutate, they become oncogenes. Mutated proto-oncogenes are known as oncogenes. They are dominant genes, right? Which means even if one gene, we have got a pair of every gene, one from dad, one from mom. In this case, because they are dominant, even if one proto-oncogene mutates and becomes an oncogene, it has the potential to cause a cancer, okay? They encode, most of the times they encode transcription factors, and they cause the cells to go on dividing. Then we have got tumor suppressor genes, and the most famous example of tumor suppressor gene is P53 gene, okay? Under normal conditions, they stop the cells from dividing. They are recessive genes, right? So if one of the genes in one of the tumor suppressor genes, remember, we get one from father, one from mother. If one mutates, then it's not a big problem. They both, on both chromosomes, they have to mutate. And only then they will affect cell division, okay? When mutated, they suffer from loss of function. They are unable to stop cell division. All right, so both of them must suffer from a loss of function. Genes that regulate apoptosis. Now, apoptosis is a very important mechanism that is there. Whenever something goes wrong in the cell, whenever a cell becomes dangerous for the neighboring cells, there are certain genes, it's known as programmed cell death, apoptosis. There are certain genes that are activated and they cause the cell to undergo apoptosis, okay? Apoptosis, programmed cell death, is initiated when the cell gets infected by a virus or there is some sort of stress or becomes precancerous. Genes that promote apoptosis are underexpressed in cancer cells. That is one of the problems in the cancers. Then genes that regulate interaction between cancer cells and normal cells. Host immune system has the capability to recognize and eliminate cancer cells, right? And natural killer cells are one of the cells that do surveillance all the time. And whenever they come across any abnormal cell, they kill it, okay? Cancer cells express genes that inhibit recognition of tumor cells by the host immune system. So it's not one mutation that's going to cause cancer, as I'll show you later on. We have to get mutations in many of these genes, okay? I'll come to that in a little while. Now, types of genetic mutations in cancers. We have got driver and passenger mutations, okay? We get a lot of mutations in the cancer. If you look at the cancer cell, examine the DNA of the cancer cells, you might find thousands of mutations, but all those mutations are not causing cancer. It's the driver mutations. They alter the functions of genes and directly contribute to development or progression of cancers, right? Passenger mutations do not affect cellular behavior and they do not contribute to development of cancer, but still they are important. Driver mutations cause cancer. Passenger mutations are just sitting there and they will go to the daughter cells, but they're not altering the function of the cells. But at a later stage, they may undergo further mutations and they may make the cancer cell resistant to treatment. That's the problem with passenger mutations. In some cancers, they cause cancer cells to become resistant to therapy, right? Down when the cells divide and um, in the daughter cells, they might cause some more mutations that make the cells resistant to cancers, all right? Point mutations, tumor suppressor genes, for example, P53, they are most commonly affected by point mutations. I've shown you point mutations previously as well, just a single nucleotide mutation. What are point mutations? Point mutations most commonly involve substitution of one base for another. For example, the GCO here has been replaced by an AT. So this is a point mutation. This has the potential to affect the function of the gene or the protein. Point mutations can alter the structure of proteins, and I've given you the example of sickle cell disease. And just to repeat that, this is the original codon which was there in the DNA. It codes for a leucine, amino acid leucine, UUA. What has happened is this U has been replaced by a C. Now, CUA also codes for a leucine. So this is known as a silent mutation. So that's not dangerous. We are going to get the same protein. But see, what has happened over here is that this U has now been replaced by a G. Now, this is known as a missense mutation. This codes for a valine. So instead of a leucine, in the protein, we will get a valine. Or we can get another type of mutation. This U has been replaced by an A. So this codes for a stop codon. Whenever we encounter this codon in the ribosome, that's the termination of the polypeptide chain. Then the polypeptide will fall off the ribosome, and that's all. That means that the protein has been completed. Now, unfortunately, if this is in the middle, then it will be a truncated protein. This is known as a nonsense mutation. This is a stop codon, so there will be no further elongation of the protein. And this is a frame shift mutation. We have got insertion of an extra U over here. So what is a frame shift mutation? I'll show you over here. You see, this is a sequence of a DNA, and we have got this, in the DNA, 3 nucleotides code for a certain protein. So we have got this CAT, CAT, all right. They are coding for a protein, okay? So this is the original sequence, CATs for a histidine. So we have got histidine. What happened was that in place of this C, there was an insertion. The C is still there, but we get an insertion. Now, what has happened? The whole frame has shifted. For example, see, we read 3 nucleotide at a time. So here we are reading 3, CAT. We will get a histidine. But here, again, we are going to read 3 nucleotide, but instead of CAT, we are going now to read ACA, right? And this will cause all the other nucleotides to be misread. For example, this is ACA. Here we will read TCA. Although the sequence after this is the same, but the frame has changed, okay? So we are reading 3 at a time. This is what is known as a frame shift mutation. And what is the consequence? Histidine. After insertion, we get a threonine and then a serine. All right, so that is what a frame shift mutation after this insertion, or it could be a deletion for that matter. All the other nucleotides will be different, and all the other amino acids are going to be different, all right? Now we have got another type of gene rearrangement, which are known as translocations. This is a very famous translocation. It is known as translocation 922. So this is chromosome number 9, and this is chromosome number 22, all right? So what happens sometimes is that both these chromosomes have broken. This chromosome 9 has broken at this point, and it has broken, chromosome 22 has broken at this point, okay? So what happened is, see, these are the broken chromosomes. Now, this portion of chromosome 22 is going to attach to chromosome number 9. And this portion of chromosome number 9 is going to attach to chromosome 22. Okay, so we get a 922 translocation, and this is known as a Philadelphia chromosome. And this is very commonly found in chronic myelogenous leukemias. Okay, we'll come to this one again later on. The translocation shown in previous figure is written as T922. Chromosome 22 is called the Philadelphia chromosome. It is present in over 90% of cases of chronic myelogenous leukemias. Another type of translocation, which is known as translocation 814, is found in over 90% of cases of Burkitt's lymphoma. You see, the pieces have broken on chromosome 8 and 14, and this piece will attach here, and this piece will attach to chromosome number 14. So we get Burkitt's lymphoma, okay? Now, just to remind you that cancer is a genetic disorder caused by DNA mutations. In this case, there is an incoming radiation, and this radiation has broken the bonds in the DNA strands, and we have lost two bases. We have lost a deletion. If this deletion occurs in a proto-oncogene, or in a tumor suppressor gene, or in a gene that controls apoptosis, then we might get genes that are non-functional or that are abnormal genes, and they might lead to cancers, all right? So deletions are common in cancers. I've shown you this deletion. So now these are single nucleotide deletions. What I'm going to show you now is deletions of parts of chromosomes. Both point mutations and deletions of specific regions of chromosomes can result in loss of tumor suppressor genes, okay? So this is chromosome number 4, and a big part of this chromosome has been deleted. So now this is still chromosome number 4, but because of this loss of this region, this is now a much shorter chromosome, okay? And this very commonly happens with T53 gene. T53 is also known as the guardian of the genome because it controls the cell cycle at a very critical point, okay? So it is the most important tumor suppressor gene lost or inactivated by deletions. And in over 50% of the cancers, we find deletions in P53 genes, okay? It is located on chromosome 17. Now, another phenomena is gene amplification. Proto-oncogenes, the genes that cause cell division, they may be converted to oncogenes by gene amplification. What is gene amplification? Gene amplification produces several hundred copies of the gene. Well, it could produce less than several hundred as well, but gene amplification basically means that many copies of that gene has been produced. Now, all those copies of the gene will be making the protein that is encoded by that particular gene, okay? So we will get, a huge quantity of those proteins, okay? Abnormally huge quantities. A change in the copy number can be detected by a special technique called molecular hybridization technique, all right? We can detect that change. We do that in breast cancer. A clinically important example of gene amplification is HER2 gene in 20% of breast cancer. So this is the normal HER2 gene on chromosome 17. What has happened is that this gene has amplified due to some mutation. Something happens, and this genes, it gets inserted into another chromosome, into this part, and what you see, this pink area is standing for this gene. So we get a very large pink area. This is known as HSR, homogeneously staining region, okay? So this shows that this gene has been amplified. It has translocated to another chromosome and it has amplified, and we can detect that. All right. Another phenomena is aneuploidy, which is present in 68% of tumors. Why am I telling you all these things? Because we look for these things when we are trying to diagnose cancers, or when we are looking at a biopsy, we want to grade the cancers. We want to see how aggressive a cancer is. So we look at all these things. The number of chromosomes is not a multiple of 23. So that is what is aneuploidy. For example, 23 is haploid genome. 46 is diploid. It is a multiple of 46. 69 is also a multiple of 23. So we call it euploidy. What is aneuploidy? It is not a multiple of 23. If the number of chromosomes is 45, 47, 70, 92, not a multiple of 23, we call it aneuploidy, okay? Then microRNAs, okay, we are now coming to the end of the lecture. MicroRNAs are single-stranded RNAs, approximately 22 nucleotide long, okay, in length. Now, why are we studying all these things? Because all these things that I'm explaining to you can cause a proto-oncogene or a tumor suppressor gene or a gene related to apoptosis. They can cause them to become non-functional or dysfunctional or mutated, all right? And that is what leads to cancers. So what are microRNAs? They function as negative regulator of genes. They inhibit gene expression by inactivating messenger RNA or cleaving the messenger RNA. So what happens is that the gene makes a messenger RNA. The messenger RNA goes out into the cytoplasm and it translates itself into a protein on the ribosomes. Now, before it could go to the ribosome, these microRNAs will attack the messenger RNA and it will make it non-functional. Uh, if the target of microRNA is a tumor suppressor gene, then overactivity of microRNA can reduce tumor suppression protein. So it will make the tumor suppressor gene non-functional. We don't want tumor suppressor genes to be non-functional. We want them to work, okay? Uh, we do not want the oncogenes to work, all right? Oncogenes are bad genes. Tumor suppressor genes are good genes, right? If the target is an oncogene, reduced production of microRNAs will lead to overproduction of oncogene product. So either underproduction of microRNAs or overproduction of microRNAs, that could lead to suppression of TSG, tumor suppressor gene, or overactivity of proto-oncogene. Sorry, not proto-oncogene. When it is overactive, it will be known as an oncogene. Downregulation of microRNAs has been found in some leukemias and lymphomas. For example, this is a long stretch of DNA and a segment over here will make a messenger RNA. This is the gene which has made this messenger RNA. Now this messenger RNA goes into the cytoplasm. But another portion of the DNA, here it makes the protein, right? Another portion of the DNA will make microRNA. MicroRNA will be complementary to some region on this messenger RNA. So here is the microRNA. And before this messenger RNA can make a protein, it will attach to the messenger RNA and it will make it non-functional, inactivated, or it will destroy it, okay? So protein assembly is eventually blocked. Now we come to epigenetic modifications. Epigenetic modifications do not cause a change in DNA sequence. So far, what we have seen is that deletions and substitutions and gene amplifications and deletion of portions of the chromosomes, they are all affecting the sequence of the DNA nucleotides, okay? But these epigenetic modifications, they do not change the sequence of DNA. For example, whatever this sequence is, they are not going to change this sequence. What they are going to do is they are going to cause methylations on cytosine residues, or they are going to cause modifications of histones. Okay, if you remember histones, we have got three or four types of histones: histone 1, histone 2A, 2B, and whatever, we have got a nucleosome around which the DNA is wound, two loops around that. Whatever it is, I'll not go into the details of genetics, but modifications in the histones and DNA methylations can cause or can disturb the functioning of the genes, okay? Epigenetic modifications cause silencing of the genes. One of the examples of DNA methylation happens in the X chromosome in females. In females, only one X chromosome is functional, or most of the genes on the X chromosome are functional only on one X chromosome. What happens to the other X chromosome? It is heavily methylated on cytosine residues, and that is why the gene on the other X chromosome, majority of the genes are silent. They are not expressing themselves, okay? Right, so DNA methylations and histone modifications, they cause loss of expression or silencing of the genes. In many cancers, tumor suppressor genes are silenced by DNA methylation as at cytosine residues. This is a cytosine, and this is a methylated cytosine. So when cytosine gets methylated, the DNA will not be able to replicate itself. The DNA polymerase will not be able to work, all right? Even the DNA-dependent RNA polymerase will also not be able to work on the DNA, all right? So this is, you see a cytosine residue and it has been methylated by a CH3 group, and you see histone tails, they are very important because whenever a DNA has to make a messenger RNA, it has to unzip, it has to open up, and that's where the histones play a part. And the tails can also get methylated or acetylated or sumoylated, many changes can take place. Now, these are known as epigenetic changes. They're not changing the sequence of the DNA, they're just changing the function of the DNA, all right? And to be more specific, they cause silencing of the gene. They do not let the gene work, okay, or express themselves. Okay, so carcinogenesis, excuse me, carcinogenesis, which means development of a cancer, it is a multi-step process. Okay, it does not, one mutation does not cause cancer. We get stepwise mutations over a long period of time. Maybe one mutation occurs at this time, the next mutation might occur after 6 years or 5 years or 2 years. The next mutation after that might occur after another 6 or 10 years. So maybe a cancer develops very fast in 1 or 2 years, or maybe it takes 20 years to develop, okay? After transformation, the cancer cells continues to acquire new mutations. This phenomena is called tumor progression. Different cancer cells may acquire different mutations independently. Now, what I'm saying over here is that once the cell becomes cancerous, after that, it will go on acquiring mutations, all right? Some combinations of mutations make the cancer cell more aggressive and difficult to treat, and some mutations that occur after transformation, transformation means now the cell has become cancer, and then it starts progressing. During the process of progression, it will acquire new mutations, or the daughter cells or the cells of the second generation, third generation, fourth generation, they might acquire new mutations that will make the tumor or the cancer more resistant to treatment, all right? When a cancer recurs after initial therapy, it most probably developed new mutations that made it less responsive to therapy. So basically, we have got a DNA repair system in every cell. Whenever there is a mutation, or whenever a nucleotide is substituted by another nucleotide, a mismatch occurs in the two chains, we have got systems that can identify that and that can correct that. That is known as DNA mismatch repair system. So what happens in the cancers is that this DNA mismatch repair system is also not working normally. So mutations go on occurring, okay? So just to explain that in a simple way, that we have got a system to repair damaged DNA. And in cancers, that system of repair also does not work normally. So we go on getting mutations once the cancer develops, okay? So here is a normal cell and some carcinogen induced a mutation. This is known as initiating mutation and initiated precursor with stem cell-like properties. So cancer cell has got certain properties which are like stem cells. You know what stem cells are? This is not a cancer cells. Then over the years or over time, it acquires new mutations, which are driver mutations, and that transforms the cell into a cancer cell, a transformed cell. Now this cancer cell will go on dividing. Its next generation will be exactly like it, but maybe the next generation develops new mutations, or after that generation, the second generation, third generation, fourth generation, now they will go on acquiring new mutations, okay? Additional mutations, emergence of subclones, and further genetic evolution. And this is how the number of mutation goes on increasing. Initially just one mutation, and the thickness indicates that the number of mutations is increasing. All right, so initially we had one clone, then one of the daughter cells acquired a new mutation, a new clone arose. Somewhere down the line, another mutation arose, a new clone. So we get many clones, emergence of subclones, okay? And then we get a genetically heterogeneous cancer. When you treat them, the drug will kill some of these cells, but some, because of different mutations, may not be killed, as I showed you in the cancer lecture, okay? So this is a benign tumor, activation of two oncogenes, but tumor suppressive genes remain normal and telomerase is remained switched off. Okay, so we have got two mutations, but two mutations are not enough to transform a cell into a cancerous cell. Now, over the years, we obtained a third mutation as well, retinoblastoma gene. Now, this is an aggressive growth, but it is still not a cancer. All right, it can still be identified by immune system. It can still undergo apoptosis. Okay, so but over the years, now we have acquired four mutations. This telomerase gene, RAS, make P53 genes. Now it has become a cancerous cell. And I'll give you another example from colorectal cancer. A single mutation is not sufficient to transform a normal cell into a cancer cell. Accumulation of multiple genetic mutations collectively give rise to full-blown cancer. So this is how it starts. The epithelium here is normal. One mutation occurred in this gene, APC gene, whatever it is, and we got some sort of abnormal growth in the epithelium. After some time, we got another mutation in a proto-oncogene. First, it was a tumor suppressor gene, adenomatous polyposis coli gene, then KRAS gene. Now the tumor is getting bigger. Then we get another mutation, DCC, deleted in colorectal cancer. These genes have strange names, and the cancer becomes more aggressive. We get a P53 mutations, 1, 2, 3, 4 mutations. And with that, we have got non-functioning mismatch repair system as well. The repair system of the DNA is not working. So the cancer becomes more and more aggressive. It grows bigger and bigger. It invades the adjacent tissues and it metastasizes, goes to the liver, the lungs, the bones, and other organs. So this shows that cancer is a multi-step process, not one gene. Many genes over time mutate, and that is what makes a cancer cell. So mutations and epigenetic changes give rise to hallmarks of cancer, okay? So the cancer cell is self-sufficient. Usually cells need growth signals, growth factors to divide. Here it doesn't need any growth factor. It is dividing itself. insensitive to anti-growth signals. We have got signals. If you withdraw growth signals, the cell will stop growing, but that does not happen in cancer cells. Limitless replicative potential, evading apoptosis. Apoptosis is not working out there. Sustained angiogenesis, the stroma. We said that stroma is very important for cancer cells. They need nutrition, they need oxygen, they need glucose, amino acid, lipids. So they need blood vessels. So they need angiogenesis, which means growth of blood vessels. They have the potential to invade, they have the potential to metastasize to other tissues. They have altered metabolism. They evade the immune system. They form certain cells that suppress the immune system, or uh, they uh, they cannot be recognized or identified by the immune system, right? So malignant tumors are more threatening than benign tumors, but both malignant and benign tumors may cause the following problems to the patients. When they grow bigger, they exert pressure on the adjacent structures. They may exert pressure on the lymphatics. They can block the lymphatics. They may exert pressure on the blood vessels. They may obstruct the blood supply to some other organ, all right? They may produce hormones, paraneoplastic syndrome, especially in lung cancers. This happens. The cancer is producing adrenocorticotropic hormone, all right? So that causes certain side effects. May cause bleeding and infections when they damage adjacent structures. They may rupture, right? Even benign tumors, they are not cancers, but they also cause problems. Malignant tumors cause cachexia, weight loss. They produce certain chemicals that cause loss of appetite and weight loss, okay? And that's all about this lecture. This slide is for your reference. These are the commonly used tumor markers that we look for when we screen the patients or a population. There are certain procedures as well, clinical procedures like colonoscopy for colorectal cancer. So that was all about, it was a little bit longish lecture, about 1.5 hours, but this will help you understand what basically cancers are. There is more to it. There are more lectures that I'll be posting. And thank you very much. That's all for this lecture.